Students, today we will discuss photosynthesis in higher plants. And what is photosynthesis? It's an enzyme regulated anabolic process of manufacture of organic compounds inside the chlorophyll containing cells from carbon dioxide and water with the help of sunlight as a source of energy. So this is 6CO2, 12H2, and here sunlight is needed and chlorophyll is needed. So with the help of this they make glucose, then again oxygen is released and water is produced. So photosynthesis is a mechanism which is followed in autotrophic nutrition. So it is who do this photosynthesis, that is autotroph. Autotrophs are doing this photosynthesis. Now photosynthesis, the total carbon dioxide available to plants for photosynthesis about 11.2 10 to the power 14 tons. Okay, so that is what we say PAR. PAR is the photosynthetically active radiation. So the plants are able to utilize only 0.2% of the solar energy received by the surface of earth. So what is the PAR value? That is 0.2% of entire solar energy that is received by the earth. Now there are number of scientists who discovered many facts of photosynthesis, and out of that the first one is Joseph Priestley. Now Joseph Priestley found that the burning candle would soon get extinguished in a closed space of bell jar. Similarly, a mouse kept in the closed space would soon get suffocated and die. Then Jan Ingenhorst, the next scientist is Jan Ingenhorst. That is Jan Ingenhorst. In his experiment with an aquatic plant, he showed that in bright sunlight, small bubbles were formed around the green parts, while in the dark, such bubbles did not form. So, and then the next scientist is von Meyer. Von Meyer, or the sacs, green plants. Now what is the sacs? Sacs, green part in plant produces glucose, which is stored as starch, and starch is the first visible product of photosynthesis. And the next scientist is Engelmann. Now this Engelmann, he split light into its components by the prism and illuminated cladophora, green alga, placed in a suspension of aerobic bacteria. He found that bacteria accumulated in the region of blue and red light of the split spectrum. So that is the action spectrum of photosynthesis. Now Blackman's law principle of limiting factors, and there is one more scientist, that is Van Nil. Now Van Nil said that studied about the purple and green sulfur bacteria, and that photosynthesis is a light dependent reaction which hydrogen from an oxidizable compound reduces CO2 from sugar. Similarly, immersion, that is an immersion effect. Robert Hill, that is evolution of oxygen occurs in light reaction. So there are several scientists who put their view, and we will discuss the process in detail with this science contribution of the different scientists. So what are the raw materials needed for photosynthesis? Carbon dioxide, then sunlight, water, minerals. So these things are needed for the raw materials. And plant must have chloroplast. Now chloroplast contain what? That is the stroma and grana. These are the two areas where the thylakoid membrane is there. Thylakoid membrane is there in the stroma and grana. So chloroplasts are the green plastids which function as the site of photosynthesis in eukaryotes. Now these chloroplasts are present in the palisade parenchyma, as we have studied in the leaf section. Chloroplast is covered by envelope of two membranes, and thylakoid membrane, the chloroplast matrix consists of the thylakoid membrane. Thylakoid membrane, when it is stacked, that means it forms the grana, where it forms the stacks of disc, and thylakoids lying outside the grana is called stroma thylakoids or intergranal thylakoids or lamellae. Now light dependent reaction occurs in grana, where the light independent reaction occurs in stroma. Now it contains four major types of complexes in this thylakoid membrane. So what are the four major complexes in the thylakoid membrane? That is photosystem 1, photosystem 2, cytochrome B6F complex and ATP synthetase complex. So four complexes are there. Photosystem 1, photosystem 2, photosystem 2, then cytochrome B6F complex and ATP synthase. So these are the four synthetase. ATP synthetase complex. So in photosynthesis, these four major complexes are there. These are present in the thylakoid membrane. Now what are the photosynthetic pigments? That is chlorophyll. So chlorophyll can be A, B, C, D and E. So 5 types of chlorophylls are there, and this chlorophyll A is found in all photosynthetic plant except bacteria. Otherwise chlorophyll A and B are majorly found in these angiospermic plants. Now these chlorophyll are associated or arranged along with the carotenoids, that is accessory pigments, and also xanthophylls. So these are accessory pigments and they are present in the reaction center. Now that is reaction center or light harvesting system, light harvesting complex, LHC. Now this LHC contains chlorophyll A, B and surrounded by all these carotenoids, and all these carotenoids trap lights and sending to chlorophyll A, and then it emits electron. So this is the LHC or the reaction center that works. So chlorophyll A and B are the major. Chlorophyll A is the reaction center whose formula is C55H72O5N4 Mg. Now, so chlorophyll, when action spectrum and absorption spectrum. Our next part of discussion is action spectrum and absorption spectrum. So absorption spectrum and action spectrum. Now absorption spectrum, this is the curve showing the amount of energy on different wavelengths of light absorbed by a substance called absorption spectrum, and that is with the help of spectrophotometer. So spectrophotometer we can see the absorption spectrum, and if you reveal absorption spectrum, you say the chlorophyll A and B that absorb maximum light in the blue and violet and red regions. So blue and red region, in this chlorophyll A and B, they absorb maximum light, and the pigments are often known as the wavelength which is absorbed to the maximum. Now action spectrum is the graphic curve depicting the relative rate of photosynthesis at different wavelengths of light. It's called the action spectrum. So action spectrum photosynthesis corresponds closely to absorption spectrum of chlorophyll A and B, showing that the latter are the main photosynthetic pigments. So action spectrum shows the rate of photosynthesis and absorption spectrum shows the light absorbed at different wavelength. Now there are the carotenoid and xanthophylls. Now they have got carotenoid. Because of this carotenoid and xanthophylls present in the reaction center, this oxidation, that is photo-oxidation, never happens. Now for photo-oxidation, the chlorophyll A will not fade. So carotenoids function as accessory pigment. They absorb radiant energy in the mid region of visible spectrum, and they protect the chloroplast constituents from nascent oxygen released from photolysis of water. Carotenoids picks up this nascent oxygen by means of their double bonds, change the same into harmless molecular state. So in this way, they act as They save the chlorophyll A from photooxidation. Now, how many what is PS1 and PS2? What is the difference between these two? PS1 and PS2. The PS1 and PS2 are what? These are the LHCs. PS1 and PS2 are the LHCs. So let's come to PS1 and PS2. This having the P700 and this is the P680. At this nanometer, that's wavelength, they absorb this water. So PS1 is a photosynthetic pigment system, and that PS1 has more chlorophyll A and chlorophyll B is less. So here PS1 is having more chlorophyll A, but here more chlorophyll B. So this is the major differences. And the light harvesting complex that absorbs the light at P700 and there it absorbs the P680 wavelength, PS2. Now both PS1 and PS2, they are very much functional, present on the thylakoid membrane. So LHC1 and LHC2, light harvesting complex 1 and light harvesting complex 2. So read NCERT. Thank you.